Welcome everyone to today's lecture on one of the core concepts in immunology, the dual arms of our immune system, innate and adaptive immunity. This presentation is part of our foundational series in the immunology curriculum and is especially relevant for biology undergraduates aiming to understand how organisms, especially humans, defend themselves against a wide range of pathogens. The immune system is a complex network of organs, cells, and molecules that work in concert to identify and eliminate threats such as viruses, bacteria, parasites, and even cancerous cells. Our primary focus today is to explore the two branches of this system, innate immunity, which provides a first and immediate response, and adaptive immunity, which offers highly specific, long-lasting protection. These systems are not mutually exclusive. They are interconnected and highly dependent on each other to function effectively. We'll explore how innate immunity sets the stage for adaptive responses, how adaptive immunity evolves during infection or vaccination, and how the two arms work together to defend us in health and disease. In today's class, we'll combine foundational theory with clinically relevant applications. By the end of this session, you should not only understand the basic mechanisms of both systems, but also how this knowledge applies to real-world scenarios like vaccine design, infection control, and immune-based therapies. All right, before we get into the nitty-gritty of immunity, let's take a quick look at what we're aiming to achieve today. These are our learning objectives, not just formal goals, but guideposts to help you track what's most important as we move through the lecture. First, by the end of this session, you should be able to clearly differentiate between innate and adaptive immunity. That means being able to look at any immune process or cell and confidently say, that's part of the innate response, or this is adaptive, it involves memory or antigen specificity. Second, you'll get familiar with the key players in each system. We're talking about the major cells like neutrophils, macrophages, dendritic cells, T cells, B cells, and the molecules they use such as cytokines, antibodies, and complement proteins. It's like learning the cast of characters in a very complex but fascinating immune drama. Third, we'll focus on how these two systems don't work in isolation. You'll learn how they interact and support each other. For example, did you know that cells from the innate system actually activate the adaptive immune response? Or that antibodies from the adaptive system can make innate cells better at killing microbes? It's truly a team effort. And finally, the fourth objective, and maybe my favorite, will apply what we've learned to real-life situations. I'll walk you through scenarios like how vaccines prepare your immune system, why autoimmune diseases happen, and how both branches of the immune system reacted during COVID-19. We'll take all this theory and see how it plays out in health and disease. So, as we go through each section, I'll remind you where we are in relation to these objectives. If you ever feel lost, just come back to these 4 points. They're the compass for today's class. Ready to dive into the first major system? Let's start with the body's fast, non-specific first responder, the innate immune system. Now, when you hear the word innate, think inborn. This part of your immune system is with you from day one. You don't need prior exposure to a pathogen for it to kick in. It's always on standby, ready to act the moment something foreign enters the body. So, what makes innate immunity special? First off, it's non-specific. That means it doesn't tailor its response to the particular pathogen. Whether it's a virus, bacteria, or fungus, the innate system has a standard set of tools it uses against all of them. It doesn't care what the invader's name is. It just wants it gone. Secondly, it's fast. This system responds within minutes to hours. Imagine cutting your finger. Within seconds, the site turns red, warm, and swollen. That's inflammation, one of the classic signs that your innate immune system has jumped into action. Third, it has no memory. Unlike the adaptive system, which remembers past infections and responds faster the second time around, innate immunity treats every threat like it's brand new. That means if the same pathogen invades a week later, the response will be the same as before, no faster, no stronger. Now, let's talk barriers. Innate immunity starts even before immune cells come into play. Your skin is a massive protective layer, dry, acidic, and constantly shedding microbes. Mucous membranes in your nose, lungs, and gut trap invaders, and are full of enzymes and antimicrobial substances. Even your tears and saliva contain lysozymes that break down bacterial walls. But if those barriers are breached, say, through a cut or inhaling a virus, then the cellular and molecular components take over. That includes phagocytes like neutrophils and macrophages, signaling proteins called cytokines, and the complement system, which we'll explore in detail soon. So, in summary, innate immunity is your body's rapid response force. It doesn't learn or evolve over time, but it buys time, holding off invaders long enough for your adaptive immune system to activate. Alright, now that we've introduced the innate immune system, let's take a closer look at the key cellular players, the immune cells that form the frontline troops of this rapid response force. Let's start with the neutrophils. These guys are the most abundant white blood cells in your body, and for good reason. Think of them as emergency responders or foot soldiers. When there's an infection or injury, they're usually the first ones on the scene. They rush to the site, detect microbes, engulf them through phagocytosis, and release toxic granules to kill them. They're short-lived but incredibly efficient. And interestingly, they form pus when they die in large numbers. Yes, pus is essentially dead neutrophils and debris from battle. Next up, macrophages. The name literally means big eaters, and they live up to it. They originate from monocytes in the blood, then migrate into tissues where they mature. Macrophages also phagocytose pathogens, but they stick around longer than neutrophils and do more. They're like the cleanup crew and the investigators combined.
Why?
Because after they digest pathogens, they can present fragments of those pathogens, called antigens, on their surface to alert the adaptive immune system. So they're not just killers, they're communicators, too. Then we have the natural killer cells, or NK cells. These are part of a fascinating class called lymphocytes, but unlike B and T cells of the adaptive immune system, NK cells act without prior sensitization. They specialize in detecting cells that are behaving abnormally, like virus-infected cells or tumor cells, and induce apoptosis, or programmed cell death. They do this by releasing perforin and granzymes, molecules that basically punch holes and activate self-destruction programs in target cells. And perhaps the most important bridge between innate and adaptive immunity, the dendritic cells. These are professional antigen-presenting cells. They patrol tissues, picking up foreign particles. When they sense danger, they migrate to the lymph nodes, where they show these antigens to T cells, essentially saying, "Hey, look what I found. Prepare to fight." Without dendritic cells, the adaptive system wouldn't get activated efficiently. So, even though they're technically part of the innate immune system, their main job is setting the stage for the adaptive arm.
So, to recap,
Neutrophils are your rapid responders, fast, deadly, and disposable. Macrophages are long-term defenders and antigen presenters. NK cells kill without needing prior training, essential for viral defense. Dendritic cells collect antigens and initiate the adaptive immune response. Each of these cells is unique, but they all work toward the same goal, identifying and eliminating anything that doesn't belong in the body. Coming up next, we'll explore the soluble factors that these cells rely on, things like cytokines and the complement system, that amplify and coordinate their attack. Let's keep going. So far, we've talked about the immune system cells, the soldiers on the ground. But just like any defense operation, cells don't act alone. They need communication tools, backup, and biochemical support. That's where soluble factors come in. Soluble factors are molecules that float around in your blood and tissues. They help coordinate the immune response, enhance killing of pathogens, and recruit more immune cells to the site of infection. The main types we'll focus on today are the complement system, cytokines, and antimicrobial peptides. Let's begin with the complement system. This is like the innate immune system's secret weapon, a cascade of more than 30 proteins that work together to detect and destroy pathogens. Once activated, complement proteins can do a few things. Opsonization. They coat the pathogen to make it easier for phagocytes like macrophages to recognize and eat them. Chemotaxis. They attract more immune cells to the site of infection. Lysis. They can directly kill pathogens by forming something called the membrane attack complex, or MAC, which punches holes in the microbe's membrane. Imagine blowing holes in a bacteria to make it leak and die. That's the MAC in action. Next up, cytokines. These are like text messages between immune cells. They're small proteins that help cells talk to each other. Different cytokines have different roles. Interleukin-1, IL-1, and tumor necrosis factor alpha, TNF-alpha, promote inflammation, making blood vessels leaky so immune cells can enter tissues. Interferons, especially type 1 interferons like IFN-alpha and IFN-beta, are released when a cell detects a virus. These interfere with viral replication and signal nearby cells to go into antiviral mode. Other cytokines recruit immune cells, ramp up production of more cytokines, or even suppress the immune response when the job is done. Think of cytokines as command signals, telling immune cells when to activate, where to go, and how to respond. However, they're potent, and when overproduced, as in a cytokine storm, they can do more harm than good. That's why their levels are tightly regulated. And lastly, we have antimicrobial peptides, like defensins and catholicitins. These are tiny proteins produced by epithelial cells and immune cells that directly kill microbes. They insert into microbial membranes and cause them to rupture. You'll find these peptides in the skin, gut, lungs, anywhere that needs an extra protective layer. So, to summarize, complement proteins amplify the attack and can directly kill microbes. Cytokines act as messengers, directing traffic and regulating inflammation. Antimicrobial peptides are tiny but mighty, direct killers of pathogens. Together, these soluble factors allow the immune system to coordinate its efforts, amplify the signal, and fine-tune the response. They're essential to the speed and efficiency of the innate immune response. In our next slide, we'll look at how the innate immune system actually recognizes invaders in the first place through structures called pattern recognition receptors. Let's go there. So now that we've seen the cells and soluble molecules involved in innate immunity, a big question comes up. How does the immune system actually recognize a threat? How do these cells know that something is foreign, dangerous, or just doesn't belong? The answer lies in special sensors called pattern recognition receptors, or PRRs. Think of PRRs as the immune system's version of radar or surveillance cameras. They don't recognize every detail about a pathogen, just the red flags. These red flags are known as PAMPs, or pathogen-associated molecular patterns. PAMPs are molecular structures that are shared among many microbes, but not present in our own cells. For example, lipopolysaccharide, or LPS, is found on the outer membrane of gram-negative bacteria. Double-stranded RNA is a common feature in viruses. Flagellin, the protein that forms bacterial flagella, is another common PMP. These are all reliable indicators that a microbe is present. Now, PRRs are germline encoded, which means they don't change from person to person. They're evolutionarily ancient and are designed to recognize these conserved microbial patterns. They're found on the surface of immune cells, inside endosomes, and even floating around in the cytoplasm. One of the most important families of PRRs are the toll-like receptors, or TLRs. There are about 10 of them in humans, each tuned to detect specific microbial components. For example, TLR4 detects LPS from gram-negative bacteria. TLR3 recognizes double-stranded viral RNA. TLR9 senses unmethylated CpG DNA, commonly found in bacterial genomes. When a PRR binds to its target PMP, it triggers a signaling cascade inside the immune cell. This leads to the activation of transcription factors like NF-kappa B, which turn on genes for inflammatory cytokines like TNF-alpha and IL-6. What happens next? The cell begins to secrete those cytokines, alerting other cells that a threat is present. It may also upregulate costimulatory molecules, preparing itself to interact with the adaptive immune system, especially T cells. Now, PRRs don't just detect pathogens, they can also recognize signs of internal damage. These are called DAMPs, damage-associated molecular patterns. So if a cell is dying abnormally, say due to trauma or a toxin, PRRs can also detect that and respond. The beauty of PRRs is their speed and reliability. They allow the immune system to recognize a wide range of pathogens very quickly without needing prior exposure. They're also one of the reasons the innate immune system kicks in so fast, sometimes within minutes. Let's summarize. PRRs detect conserved microbial features, known as PAMPs. They trigger inflammatory responses via signaling pathways. TLRs are the best-known PRRs, and they're located on cell surfaces and inside cells. PRRs also detect DAMPs, helping the immune system respond to internal threats, like damaged tissues. Next, we'll tie together everything we've learned so far and look at a summary of innate immunity to see how these barriers, cells, and molecules all come together during an immune response. Okay, before we shift gears and explore the adaptive immune system, let's take a moment to pull everything together and summarize what we've learned about the innate immune system. We started with the basic idea that innate immunity is immediate, nonspecific, and present from birth. It's the first responder system, acting quickly to detect and eliminate foreign invaders. And while it's not tailored to specific pathogens, it's incredibly effective at controlling infections early on. Let's go over the key features. First, we talked about speed. Innate immunity kicks in within minutes to hours of an infection. There's no delay while the system figures out what it's dealing with. It's more like, see invader? Attack now. This is in stark contrast to the adaptive system, which can take several days to activate fully. Second, it's nonspecific. That doesn't mean it's careless. It just means it doesn't target specific strains or viral subtypes. Instead, it recognizes common structures shared among groups of pathogens. This is why PRRs and PAMPs are so important. They let the innate system sense danger quickly and efficiently. Third, there's no memory. If you're infected with the same virus twice, the innate immune system will respond exactly the same way both times. It doesn't remember the invader. That job belongs to the adaptive immune system, which we'll dive into shortly. Now, let's revisit the components. We discussed the physical barriers, like the skin, mucous membranes, and secretions that block entry of pathogens in the first place. Then we looked at the cellular players. Neutrophils, the fast, short-lived phagocytes that swarm to infection sites. Macrophages, longer-lasting cells that not only engulf microbes, but also alert the adaptive system. Natural killer cells, the innate lymphocytes that can detect and kill infected or abnormal cells without prior sensitization. Dendritic cells, the all-important bridge between innate and adaptive responses, capturing antigens and presenting them to T cells. We also covered soluble mediators, including the complement system, which tags pathogens, recruits immune cells, and can directly lyse invaders. Cytokines, which act as signaling molecules to promote inflammation and cell recruitment. Antimicrobial peptides, like defensins, that directly kill bacteria and fungi by disrupting their membranes. And finally, we explored pattern recognition receptors, especially toll-like receptors, which detect common microbial patterns and launch inflammatory responses. These receptors are the sensors that allow innate immunity to recognize pathogens and act fast. So to wrap up, innate immunity is your first line of defense. It's fast, general, and powerful, but it doesn't improve with experience. Its greatest strength is its speed and reliability. And perhaps most importantly, it sets the stage for the more targeted and long-lasting adaptive immune response. Coming up next, we're going to shift focus and explore that adaptive side, the part of the immune system that learns, remembers, and adapts. Let's dive into what makes adaptive immunity so powerful and so different. Alright, now that we've covered the fast and furious world of innate immunity, let's take a step into something a little more specialized, a bit more strategic, the adaptive immune system. If innate immunity is the body's rapid response team, adaptive immunity is the elite tactical force. It doesn't rush in blindly. It studies the invader, crafts a custom response, and most importantly, it remembers the encounter for next time. Let's break this down. Adaptive immunity is specific. Unlike innate immunity, which recognizes broad microbial features, adaptive immunity targets unique antigens, specific molecular fingerprints found on pathogens. These can be pieces of viral proteins, bacterial toxins, or even mutated self-proteins in cancer. It's also highly regulated and slower to respond. When the adaptive immune system is first exposed to a new pathogen, it takes a few days to really get going. Why the delay? Because it's busy doing something incredible, selecting and activating specific lymphocytes, T and B cells, that match the exact antigen it encountered. This process is called clonal selection and expansion. It ensures that only the lymphocytes with the right receptor are activated and multiplied. Now here's the best part, memory. Once the adaptive immune system has responded to a pathogen, it keeps a record. Memory T and B cells stick around, often for years, ready to respond faster and stronger if that same pathogen returns. This is the fundamental principle behind vaccination. Expose the immune system to a harmless form of the pathogen, and it will remember how to defeat the real thing. Let's briefly introduce the two arms of adaptive immunity. The humoral response is mediated by B cells, which produce antibodies that circulate in the blood and neutralize extracellular pathogens. The cell-mediated response is driven by T cells, which include helper T cells, CD4+, that coordinate the immune response, and cytotoxic T cells, CD8+, that directly kill infected or abnormal cells. It's also worth noting that adaptive immunity requires antigen presentation. The innate system doesn't just fight off infection, it hands off the baton. Antigen-presenting cells like dendritic cells display processed antigens on their surface using MHC molecules. This is how T cells recognize what to attack. So again, you see the tight partnership between innate and adaptive immunity. To summarize, adaptive immunity is slow at first, but highly specific to each pathogen. It's mediated by B and T lymphocytes. It generates immunological memory, providing long-lasting protection. And it relies on the innate system for activation and direction. In contrast to the generalized, immediate innate response, adaptive immunity is personalized, precise, and persistent. Now that we've introduced the adaptive immune system, it's time to meet the main characters, the cells that do the heavy lifting. These are your B cells and T cells, collectively known as lymphocytes. Let's start with T cells, which mature in the thymus. That's what the T stands for. There are two main types you'll need to know. First, we have the CD4+ helper T cells. These cells don't kill pathogens directly, but they're absolutely essential. Think of them as commanders on the battlefield. Once activated, they release cytokines, those chemical signals we talked about earlier, that orchestrate the entire immune response. Depending on the cytokines they produce, CD4+ cells can take on different roles. Th1 cells help activate macrophages to destroy intracellular bacteria. Th2 cells are involved in activating B cells and promoting antibody production. Th17 cells specialize in recruiting neutrophils and defending against fungi. And T regulatory cells, TREGs, act as peacekeepers, suppressing immune responses and preventing autoimmunity. Then we have the CD8+ cytotoxic T cells. These are your killers. Once activated, they seek out cells infected with viruses or transformed into cancer cells. They recognize antigens presented by MHC class I molecules, which are found on virtually all nucleated cells. When a target is identified, CD8+ T cells release perforin and granzymes to induce apoptosis. That's a clean, controlled cell death. This is how your body gets rid of infected or abnormal cells without causing collateral damage. Now, let's talk about B cells. These mature in the bone marrow. That's where the B comes from, and their main job is to produce antibodies. Each B cell carries a unique receptor on its surface that can bind to a specific antigen. When a B cell encounters its matching antigen and often receives help from a CD4+ T cell, it becomes activated and transforms into a plasma cell, which acts as an antibody factory. Some activated B cells also become memory B cells, ready to respond even faster the next time that same antigen appears. So, in short, CD4+ T cells help coordinate the response, activating other immune cells and directing traffic. CD8+ T cells are cytotoxic. They kill infected or damaged cells directly. B cells are the antibody producers, providing humoral immunity. One fascinating thing about both T and B cells is how they generate their receptors. Through a process called V-D-J recombination, these cells shuffle segments of DNA to create millions of different antigen receptors. This means your adaptive immune system can recognize virtually any pathogen it might ever encounter, even ones that have never existed before. It's like having an immune system that writes its own code on the fly. This receptor diversity is what makes adaptive immunity so powerful, but it also explains why it takes a few days to mount a response. Your body has to find and activate the few cells that recognize the new invader. So, let's wrap this up. Adaptive immunity depends on highly specialized lymphocytes. T cells are involved in killing and coordination. B cells handle antibody production. And the diversity of their receptors allows for incredible specificity. Now that we've introduced B cells, let's take a closer look at what they actually do once they're activated. This brings us into the realm of humoral immunity, which, by the way, comes from the word humor, meaning body fluid. So, humoral immunity refers to the antibody-mediated defense that operates in the blood, lymph, and other extracellular fluids. Let's start at the beginning. A naive B cell circulates throughout the body, each one equipped with a unique B cell receptor, or BCR. This receptor is basically a membrane-bound form of an antibody. Each B cell is specific to one particular antigen, one molecular shape. When a B cell encounters that matching antigen, either floating freely or presented by a helper T cell, it gets activated. And what happens next? The B cell undergoes clonal expansion. It multiplies, creating a whole army of identical B cells. Most of these turn into plasma cells, which are essentially antibody factories. These cells pump out thousands of antibodies per second, all designed to bind that specific antigen. Now, let's talk about the antibodies themselves, also known as immunoglobulins, or Ig for short. They're Y-shaped proteins, and they work by neutralizing pathogens, meaning they bind to viruses or toxins and block them from entering host cells. Opsonizing pathogens, which means coating them to make them easier for phagocytes like macrophages to eat. Activating complement, which we learned earlier, helps punch holes in pathogens or flags them for destruction. There are 5 main classes of antibodies. IgM is the first one produced during an initial infection. It's large, forms pentamers, and is great at activating complement. IgG comes next. It's smaller, more versatile, and provides long-term protection. It's the most abundant antibody in blood and can cross the placenta to protect newborns. IgA is found in mucosal areas like the respiratory and gastrointestinal tracts and in secretions like saliva, tears, and breast milk. IgA is the one we blame for allergies. It binds to mast cells and causes histamine release. But it's also essential in defending against parasites. IgD is the least understood. It plays a role in B cell activation, but isn't well characterized. One key feature of B cells is that antibodies work outside the cells. Unlike cytotoxic T cells that hunt infected cells, antibodies float freely in the bloodstream and tissues, tagging extracellular invaders for destruction. This makes them perfect for fighting bacteria, viruses, and toxins before they enter cells. Also, during the immune response, B cells undergo class switching, changing the type of antibody they produce, like switching from IgM to IgG, and affinity maturation, which fine-tunes how tightly the antibody binds the antigen. These processes make the response stronger and more effective over time. Let's sum it all up. Humoral immunity is mediated by B cells and the antibodies they secrete. It defends against extracellular pathogens in fluids like blood and mucus. Antibodies neutralize, opsonize, and activate complement. There are 5 antibody classes, each with specific roles and locations. This system is the basis of many vaccines. By exposing the body to a harmless version of a pathogen, we train B cells to create memory and produce protective antibodies long-term. All right, now that we've talked about how B cells and antibodies handle threats outside our cells, it's time to switch gears and talk about the part of the adaptive immune system that handles the threats inside our cells. This is cell-mediated immunity, and it's the domain of the T cells. So, why do we even need the system? Well, some pathogens like viruses, intracellular bacteria, and even cancer cells hide inside our own cells. Antibodies can't reach them there. That's where T cells, particularly cytotoxic T lymphocytes, or CD8 plus T cells, step in. Let's walk through how it works. When a cell becomes infected by a virus, or turns cancerous, it starts to display small pieces of abnormal proteins on its surface using a molecule called MHC class I. This is like waving a tiny flag that says, something's wrong inside. Every nucleated cell in your body has MHCI. It's a universal alert system. CD8 plus T cells constantly patrol the body, checking these MHCI flags. When they find one that displays a non-self or altered peptide, they become activated. Once activated, they release perforin, which creates pores in the target cell's membrane, and granzymes, which enter the cell and trigger apoptosis, a programmed cell death process. It's precise, clean, and highly controlled. No unnecessary inflammation. Just surgical elimination of the threat. Now, let's talk about the CD4 plus helper T cells, which we introduced earlier. These cells are activated when they recognize antigens presented on MHC class II molecules, which are found only on professional antigen-presenting cells, like macrophages, dendritic cells, and B cells. Once activated, CD4 plus cells don't kill anything directly. Instead, they act as coordinators of the immune response. They release different cytokines that help activate B cells to produce antibodies, recruit and activate macrophages to destroy pathogens, and guide CD8 plus T cell responses. There are even different subsets of CD4 plus cells, each suited for different types of threats. Th1 cells help fight viruses and intracellular bacteria. Th2 cells promote antibody production against parasites and allergens. Th17 cells recruit neutrophils to fight extracellular bacteria and fungi. And T-regulatory, TREG, cells suppress immune responses to prevent autoimmunity and excessive inflammation. Together, CD4 plus and CD8 plus T cells provide a powerful, targeted, and versatile defense system against infections and diseases that antibodies alone can't handle. And just like B cells, T cells also generate memory cells. These stick around long after the infection is cleared, enabling a faster, stronger response if the same pathogen returns. Here's a summary. CD8 plus cytotoxic T cells recognize antigens on MHCI and kill infected cells. CD4 plus helper T cells recognize antigens on MHC2 and activate other immune cells via cytokines. Both cell types undergo clonal expansion and form memory cells. Cell-mediated immunity is crucial for clearing viral infections, intracellular bacteria, and cancer cells. This system doesn't just help fight disease. It also plays a role in transplant rejection, autoimmune disorders, and is the target of many new immunotherapies, especially in cancer treatment. We've now talked about how the adaptive immune system identifies specific threats and mounts highly tailored responses. But one of its most remarkable features, and what truly sets it apart from innate immunity, is its ability to remember. This is what we call immunological memory, and it's the reason why you don't get chickenpox twice, and why vaccines work. So, let's explore how this works. After an infection or a vaccination, most of the activated B and T cells complete their mission, then undergo apoptosis. They die off to prevent an ongoing, unnecessary immune response. But not all of them. A select group of these cells become memory cells. There are memory B cells, memory CD4 plus T cells, and memory CD8 plus T cells. These cells don't fight immediately. Instead, they stick around, sometimes for years or even decades, patrolling quietly, ready to respond if the same antigen shows up again. Let's say you're exposed to a virus for the first time. It takes about a week for your adaptive immune system to mount a full response, activating the right B and T cells, expanding them, and producing enough antibodies and killer T cells to control the infection. That's called the primary response. Now fast forward a few months. The same virus shows up again. But this time, your memory B cells quickly recognize the antigen and begin producing high-affinity antibodies faster and in greater quantity. Your memory T cells also spring into action almost immediately. This is the secondary immune response, and it's faster, stronger, and more efficient. You can even visualize this with a classic immunology graph. The first response is slower, mostly IgM. The second response peaks faster and is dominated by IgG, with higher affinity and longer duration. This is the whole concept behind vaccination. You give the immune system a safe, inactivated version of a pathogen, like a weakened virus or a bit of its protein, so it can mount a harmless primary response and store that information in memory cells. Then, when the real pathogen appears, the body treats it like an old enemy and neutralizes it before you even feel symptoms. It's also worth noting that memory can sometimes wane over time, which is why booster shots are needed. Boosters re-stimulate memory cells and increase antibody levels before they drop too low. And in some cases, like influenza, the virus changes its antigens frequently, so previous memory isn't fully protective. That's why the flu vaccine changes each year. Here's a quick summary. Immunological memory is long-term protection generated after infection or vaccination. Memory B and T cells respond faster and more robustly upon re-exposure. The secondary response is more efficient and often prevents illness altogether. This is the biological basis of vaccination and long-term immunity. In short, memory is what turns your adaptive immune system into a personalized, lifelong defense system. Before we move into comparing the two branches of immunity side by side, let's pause for a moment and wrap up what we've learned about adaptive immunity. It's a lot, and it's powerful. Let's start with the big picture. Adaptive immunity is all about specificity, precision, and memory. It doesn't act instantly like the innate system, but when it does act, it's highly targeted, designed to destroy a particular pathogen, not just any invader. One of its most incredible strengths is its ability to recognize and respond to almost any antigen.
How?
Through a process called somatic recombination, or V-D-J recombination. B and T cells rearrange their DNA to create unique antigen receptors. This gives rise to an enormous diversity, literally millions of different receptor types. So even if your immune system has never seen a particular pathogen before, odds are some B or T cell exists that can recognize it. Now, the adaptive system consists of two arms. The humoral arm, driven by B cells, which produce antibodies that circulate in the blood and lymph, neutralizing pathogens and marking them for destruction. The cell-mediated arm, driven by T cells, specifically CD8+ cytotoxic T cells that kill infected or cancerous cells, and CD4+ helper T cells that coordinate the entire immune response through cytokine signaling. We also talked about memory, perhaps the adaptive system's biggest advantage. Once exposed to a pathogen, it remembers. Memory B and T cells ensure that if that same pathogen returns, your body responds faster, stronger, and often without symptoms. What else did we learn? That B cells can switch antibody classes and undergo affinity maturation to improve their response. That T cells require antigen presentation. CD4+ cells recognize peptides on MHC2 from professional antigen-presenting cells, and CD8+ cells recognize peptides on MHC1 from all nucleated cells. That helper T cells have specialized subtypes, Th1, Th2, Th17, Tregs, tailored to different pathogens or immune scenarios. And that cytotoxic T cells use very targeted killing mechanisms, releasing perforin and granzymes to destroy infected cells without harming their neighbors. Let's not forget how adaptive immunity relies on innate immunity to get started. Dendritic cells from the innate system present antigens. Cytokines from innate cells help shape the direction of adaptive responses. So even though adaptive immunity is highly specialized, it's not self-sufficient. It needs that innate wake-up call to get going. Here's the summary in a nutshell. Highly specific to antigens. Slower to start, but stronger and more refined. Generates memory, providing long-lasting protection. Driven by lymphocytes, B cells, antibody producers, and T cells, coordinators and killers. Constantly shaped by antigen presentation and cytokine signaling. This system is what modern vaccines leverage. It's what immunotherapies harness. And it's the reason you're protected from diseases you had years ago, or never had at all. Now it's time to bring everything together and do a side-by-side comparison of the two major arms of the immune system, innate immunity and adaptive immunity. Think of this as the ultimate checkpoint in your understanding. We've gone through both systems in depth. Now let's highlight their key differences and, more importantly, how they work together. Let's start with the response time. Innate immunity is immediate. It's active within minutes to hours of an infection. It's like your body's default setting, always on, always scanning. Adaptive immunity, on the other hand, is delayed. It can take days to kick in during a primary exposure because it needs time to identify the invader, activate the right lymphocytes, and mount a targeted attack. Next is specificity. Innate immunity is nonspecific. It recognizes general features common to many pathogens, like bacterial cell walls, viral RNA, or fungal sugars. Adaptive immunity is highly specific. It identifies unique antigens, specific pieces of one pathogen that no other cell has. How about memory? The innate system doesn't remember. It reacts the same way each time. The adaptive system develops memory. It responds faster and stronger upon re-exposure. This is the cornerstone of long-term immunity and vaccination. Now, let's look at the cell types involved. Innate immunity relies on cells like neutrophils, macrophages, dendritic cells, and NK cells. These cells are ready from birth and recognize conserved pathogen features using pattern recognition receptors, PRRs. Adaptive immunity is powered by B cells and T cells, which use antigen-specific receptors generated through gene rearrangement. Each B or T cell is unique in the antigen it recognizes. Regarding receptor diversity, innate immune receptors are germline encoded. That means they're hardwired in your DNA and inherited. They don't change. Adaptive immune receptors are somatically recombined. B and T cells rearrange their DNA to create receptors that are unique and diverse. Millions of combinations exist within a single individual. Let's also consider evolutionary age. Innate immunity is ancient, found in nearly all multicellular organisms, from insects to humans. Adaptive immunity is more recent, only found in vertebrates like mammals, birds, and fish. And finally, let's talk about cooperation, because these two systems aren't separate silos. They're deeply interconnected. Innate cells like dendritic cells and macrophages activate adaptive immunity by presenting antigens and secreting cytokines. Adaptive components, like antibodies, enhance innate processes, such as opsonization and complement activation. Cytokines from T helper cells also influence the behavior of innate cells. So really, instead of seeing these two systems as opposites, think of them as teammates. The innate system is the fast scout, and the adaptive system is the precision strike team, both essential to winning the battle against infection. Now that we've compared innate and adaptive immunity side by side, let's talk about something even more important: how these two arms of the immune system work together. Because in reality, they're not separate systems. They're deeply integrated, and they constantly communicate. Let's go back to the analogy of a defense team. The innate immune system is the first responder. It detects the problem, holds the line, and raises the alarm. But it also acts like an intelligence officer, collecting information about the invader and sending it to the adaptive immune system, which acts like a specialized strike force. They don't work in isolation. They depend on each other. The best example of this coordination is the role of dendritic cells. These are technically part of the innate immune system, but they're also the key to activating adaptive immunity. When dendritic cells encounter pathogens, they engulf the invader, process it, and display antigen fragments on MHC molecules. Migrate to lymph nodes, where they interact with naive T cells. Present antigens to the T cells, delivering the message: this is what the enemy looks like. Activate your forces. Without dendritic cells and other antigen-presenting cells, your T cells would never know where to go or what to attack. So the adaptive response literally depends on innate signals. It doesn't stop there. The cytokines produced by innate cells during early infection also help direct the type of adaptive response.
For example,
if dendritic cells secrete IL-12, They push helper T cells to become Th1 cells, promoting macrophage activation. If they secrete IL-4, they promote Th2 responses, which help B cells produce antibodies. This is how the innate system informs the adaptive system about the nature of the threat, virus, bacteria, parasite, etc., so the response can be tailored appropriately. Now, let's flip it. Adaptive immunity also enhances innate responses. Antibodies, produced by B cells, can bind to pathogens and opsonize them, making them easier for macrophages to eat. Certain antibody subclasses, like IgG, can trigger the complement cascade, which is an innate mechanism for lysing microbes. CD4 plus T cells can activate macrophages, making them more aggressive killers. Memory cells formed during adaptive responses can even influence how innate cells behave during future infections. This feedback loop is what makes immune responses so effective. It's not just two systems working in parallel, it's a dynamic conversation. Information flows back and forth, from innate to adaptive, via antigen presentation and cytokines, and from adaptive to innate, via antibodies and helper signals. Let's take an example. During a viral infection, innate sensors detect viral RNA and trigger interferon release. Dendritic cells present viral peptides to CD8 plus T cells. These cytotoxic T cells then patrol the body and kill virus-infected cells. Meanwhile, B cells produce antibodies that neutralize free-floating viral particles. The innate system detects, the adaptive system clears, and both systems enhance each other. To summarize, innate immunity activates and shapes adaptive immunity. Adaptive immunity enhances and refines innate responses. Together, they create a coordinated, flexible, and powerful immune defense. Now that we've covered how the innate and adaptive systems work together, let's zoom out and see how all of this plays out in the real world. Because understanding immunity isn't just about acing an exam, it's about understanding how your body responds to real threats and how that knowledge is used in medicine, public health, and clinical decision-making. Let's start with the most familiar application, vaccination. Vaccines are designed to train the adaptive immune system without causing disease. When you get vaccinated, you're usually being exposed to a weakened, inactivated, or fragmented version of a pathogen. This is enough to trigger an immune response. B cells produce antibodies, T cells get activated, and most importantly, memory cells are formed. This means that when your body encounters the real pathogen in the future, it already has the tools to neutralize it, quickly and efficiently. The innate system still responds, but the adaptive system can leap into action far faster, thanks to immunological memory. Vaccines are one of the greatest public health achievements in history, and they only work because of the precise memory function of adaptive immunity, triggered and shaped by innate signals. Now, let's talk about autoimmune diseases. In conditions like type 1 diabetes, rheumatoid arthritis, or multiple sclerosis, the adaptive immune system goes rogue. It starts attacking the body's own cells, mistaking them for pathogens. This happens because of a failure in self-tolerance. Normally, B and T cells that recognize self are eliminated or silenced during development. But when that fails, the immune system can become its own worst enemy. Innate immunity also plays a role here. Chronic activation of innate sensors, like PRRs responding to danger signals, can worsen inflammation and trigger autoimmunity. So again, it's a partnership, but when the balance is off, disease happens. What about sepsis? Sepsis is a life-threatening condition caused by an uncontrolled innate immune response, usually to a bacterial infection. Here, the immune system overreacts. Massive amounts of cytokines are released in what's called a cytokine storm. Blood vessels become leaky, organs start to fail, and unless it's treated quickly, it can be fatal. It's an example of the innate immune system going into overdrive, without enough regulation from the adaptive arm. Understanding this has helped clinicians develop more targeted therapies and supportive care strategies. Now, let's look at a modern example we've all lived through, COVID-19. With SARS-CoV-2, the virus that causes COVID-19, the innate immune response is the first line of defense. In people with a strong early response, especially production of interferons, the infection is often mild or even asymptomatic. But when the innate system is delayed or weak, the virus spreads rapidly, and the adaptive system is left playing catch-up. This lag can lead to severe disease and, again, cytokine storms that cause widespread inflammation. Vaccines, especially mRNA vaccines, were developed to train the adaptive immune system to recognize the viral spike protein. This meant vaccinated individuals could mount a rapid adaptive response, limiting the spread and severity of the infection. So, to summarize, vaccination leverages adaptive memory and innate instruction. Autoimmunity involves adaptive misfiring and sometimes innate overactivation. Sepsis is a failure of innate regulation. COVID-19 showed us the delicate timing and cooperation required between both systems. These aren't just textbook concepts, they're lived experiences, clinical challenges, and public health strategies. Understanding the immune system empowers us to make better decisions, develop better treatments, and ultimately save lives. Thank you all so much for your time and focus today. I hope you now have a solid understanding of both innate and adaptive immunity, how they differ, how they work together, and how this knowledge applies in the real world. If you found this class helpful, please let me know. I love hearing feedback from students. And if you have any questions, feel free to reach out during office hours or drop me an email. I'm always happy to help clarify things or point you toward more learning resources. And one last thing, make sure to subscribe to the channel to follow the course updates. We'll be covering more key immunology topics soon, like hypersensitivity, immune tolerance, vaccination strategies, and immune evasion by pathogens. So hit that subscribe button, turn on notifications, and stay tuned for future lectures that will build on what we discussed today. Once again, thank you, and keep questioning, keep learning, and keep exploring the amazing world of immunology. See you next time.